Hi everyone, I am going to make a video about digestion. So I'm going to review um, carbohydrate, lipid, and protein digestion, and then I'll finish up talking about some nutrition issues and ending with some clinical correlations that connect to the digestive system. So, let's just remind ourselves, big picture, that the food that we eat comes in compounds that are typically too large for us to immediately extract nutrients from, and they are often water insoluble. So we have two issues there that we need to address through digestion, and that's why we are going to need both physical and chemical digestion, because we're going to need to break physically some of these compounds down into smaller pieces, and then we're also going to chemically digest them into smaller pieces so that ultimately those nutrients can be absorbed across the epithelial lining of the digestive tract. And the ones we're going to focus on, as I mentioned, are going to be carbohydrates, lipids, and proteins. We don't really focus on nucleic acids since that's not a primary goal of the digestive system. So real quick, let's just remind ourselves of some things that we should probably already know. But this is a good introduction and a review, whichever way it works for you. So let's just remind ourselves that in the mouth, we are going to have food. Some food particles can be dissolved if they're water soluble by being exposed to saliva, but we're also going to have mechanical digestion by the teeth and the tongue. And then also we are going to have some enzymes in the mouth, like amylase, but also lingual lipase, for example, that will begin the process of breaking down carbohydrates and lipids. It's certainly not completed in the mouth, but the process starts there. So when the food in the form of a bolus slides down the esophagus and ends up in the stomach, we're going to continue that physical and chemical breakdown. Because remember, the stomach has 3 layers of muscle, so it can squeeze and churn and help continue to mix and break physically down the food that we've eaten. But also, the stomach is going to have stomach acid, hydrochloric acid, and also enzymes that are going to start chemically digesting that food. Now, that means that the food in the stomach, it's partially digested now, so we're going to call it chyme, but it's also very acidic as a result of that hydrochloric acid. So when it leaves the stomach through the pyloric sphincter of the stomach and enters the duodenum, we now have this very acidic chyme. So we now have a couple more things we need to do. We still need to continue breaking down those food molecules so that we can absorb the nutrients, and that's going to be guided in large part by enzymes coming from the pancreas that are going to be stimulated for release by hormones like CCK and secretin. But we also have the issue of we do have that super acidic chyme, and we do need to bring that pH back up and get it closer to neutral so that the acidity of the chyme doesn't damage the small intestine. So it's going to continue to move through the small intestine, and we're going to find that in the jejunum of the small intestine, we're going to have the bulk of nutrient absorption, although quite a bit of absorption also happens in the duodenum. By the time we get into the large intestine, so that would be, remember, the last part of the small intestine is the ileum. It joins the large intestine at the cecum, which is also located in the area where the appendix can typically be found hanging off, which is right over here. In the large intestine, we are going to see that the biggest thing that's happening there is the absorption of water. We want to dry out that fecal matter, but we are going to have a little bit of nutrient absorption, in particular for some fat-soluble vitamins, because we actually need the help of bacteria in our large intestine to do that. And then, of course, the large intestine is going to be responsible for excreting waste material. And then thinking about the accessory organs of the digestive system. Remember, all those places, some things in the stomach, more in the duodenum, lots in the jejunum, and a little bit in the large intestine. All those nutrients, we need to get them somewhere, right? The whole point was to get them out to the cells of our bodies that need them. So we're going to collect all of those nutrients in the capillary network that's found in the mesentery surrounding the digestive tract, and all of those nutrients are going to be transported to the liver via the hepatic portal vein, and then the liver will absorb all of these nutrients and sort of do them up. In some cases, it packages them up or it modifies or alters them in some way and then releases them out to the rest of the body as needed. And so cells in the peripheral tissues can then absorb those nutrients. So this little picture, it might not look like that much or it might not seem that important, but it really gives you this great overview of all of the things that we've been talking about. And it sort of relates the anatomy to the physiology. So the basic structures to the physiology or the processes that the digestive system is carrying out. Okay, now for carbohydrate digestion, and I am going to go through these things relatively quickly because we have covered them previously in class, but carbohydrate digestion is important to us because it's the preferred substrate for catabolism of inside cells to make ATP in the mitochondria, because glucose is really easy for your cells to work with, and that's why it likes it. So some of the major players or enzymes that are going to help us break down carbohydrates are going to include salivary amylase that's in the mouth, pancreatic alpha amylase that's coming from the pancreas and entering the duodenum. There's a whole bunch of others like maltase and sucrase and lactase. These are all going to break down disaccharide sugars. And again, the goal here is we're going to take more complex sugars, they're either disaccharides or bigger, and we're going to break them down into simple sugars like glucose because those are much more readily absorbable across the epithelial cells of the digestive tract. The glucose that we absorb, those simple sugars that we absorb, will ultimately end up in the liver. The liver will most likely convert those the glucose into glycogen, which remember is just a whole bunch of glucoses covalently bonded together, and that allows us to store that glucose for longer periods of time and not have it floating around in the blood, elevating our blood sugar levels. But when the body needs those simple sugars, then glycogen can be broken down by the liver, and that glucose can be released out into the blood supply for cells. So cells can pick up glucose in response to insulin, which of course connects back to GIP, which helps stimulate the release of insulin. There is a whole bunch of carbohydrates, though, that we can't digest. We don't have enzymes that allow us to break them down. And so those are indigestible carbs, and that includes cellulose. Cellulose is a very, I don't know, have words right now. Cellulose is the structural carbohydrate that makes up plants. So when you eat leafy greens, for example, there's a lot of cellulose because it's the structural component for them, and we can't digest that. So that's fiber. And if we kind of blow up this picture over here, again, it's kind of repeating some of those things from the first slide, but then it's also getting into the details specific to carbohydrates. So again, we can see we've got salivary amylase that's going to start breaking down carbohydrates in the mouth. That's going to continue into the stomach. And once that chyme moves into the duodenum, we can see that we're going to have secretin stimulating the release of buffers to help bring up those pH levels. We're going to have cholecystokinin or CCK releasing additional buffers and enzymes from the pancreas, including pancreatic alpha amylase, which is just like salivary amylase. It's going to help break down carbohydrates into simple sugars. We are also going to see that the arrival of chyme in the duodenum is going to trigger the release of GIP, which will stimulate the release of insulin by the pancreas because we're about to get a whole bunch of glucose headed into our blood. And so we want to make sure that we can regulate that glucose level. And that's the job of insulin, is to regulate blood sugar levels. Again, here we can see the indigestible carbohydrates are going to pass through the entire digestive tract. They may increase gas in the intestinal tract. And then over here in these little pictures, it's just showing you how the liver can take that glucose and lock it up into glycogen or break down glycogen and release it as glucose. And in this little picture, we're seeing how glucose can be absorbed across the epithelial cells lining the digestive tract. And they're just pointing out that some of those enzymes I mentioned earlier will break down disaccharides into simple sugars because those are easy to move across the epithelial cells and ultimately into the blood supply so that they can go to the liver and the liver can process them. So for some people, studying those diagrams can really help you develop an understanding. That brings us to lipid digestion, which is a little more complex. It's a little bit harder for us to break down lipids. So we're going to start the process with lingual lipase in the mouth, and it will start breaking down triglycerides that we eat into monoglycerides and fatty acids. But it only does about 20%. It's not super effective. So once we get into the stomach and then into the duodenum, we're going to have additional breakdown of those lipids. The release of bile because of CCK is going to help take large lipid droplets and break them down into tiny droplets, which in turn will allow pancreatic lipases, which are very similar to lingual lipases. They'll have better access to those lipids now because they're smaller, so they'll be able to break those lipids down into even smaller droplets or smaller pieces that we now call micelles, because those micelles will be a little bit of lipids surrounded by some of the bile salt complexes from the bile that's been released from the gallbladder that was made by the liver. And then those micelles will actually sort of edge up against the epithelial cells, and when they do that, the epithelial cells will be able to convert those micelles into something we call chylomicrons, because micelles are not water soluble, they're still lipid soluble, but in order to put these things into the blood, we need them to be water soluble. So the epithelial cells will convert the micelles into chylomicrons, which another name for those is lipoproteins. So essentially we're going to take the lipid and the epithelial cell is going to wrap it with a protein coat, which makes it water soluble. So now it can enter the blood and it can go to the liver and the liver can process it. And what the liver is going to do with those chylomicrons is convert them to LDLs and HDLs. These are types of cholesterols. And so if we look at this image here, and there is a bigger version of this image with all of the steps in your book, but I'm just focusing on what happens to the liver, what happens in the liver here to these. So when these chylomicrons arrive in the liver is going to convert them into VLDLs, which stands for very low density lipoproteins, and then also low density lipoproteins. So the VLDLs we don't care that much about, but they're going to go off to the muscle and the adipose tissue for long-term storage or for use as a source of energy in muscle. So these VLDLs really don't cause any issues for us. They get used and, we don't really want too much fat stored in our adipose tissue, but it's at least locked up. Now, the LDLs, on the other hand, have a little bit more of an interesting story. The LDLs are going to go out to the peripheral tissues and they're going to be taken up by cells, and the cells will use these LDLs for things like renewing their cell membranes, whether that's the actual cell membrane or membranes within the cell that form like organelles, for example. But some of these cholesterols are also going to be used to make hormones. Remember, like testosterone and estrogen both have cholesterols in them. So the cells will use those up. But if more cholesterol, more LDL is coming to the cells than what they need, then the cells will release that excess cholesterol back into the blood. Now, one of the other things that your liver makes is HDLs, and HDLs are really good at picking up excess cholesterol. They grab that excess cholesterol and they're going to bring it back to the liver. And what we hope kind of happens is that the liver processes it and that extra cholesterol is excreted with the bile salt, so it actually passes out in fecal matter. And so we get rid of that extra cholesterol. But if you don't have enough HDLs to pick up the excess cholesterol, and of course that goes the other way, if you're consuming so much cholesterol that you have so much excess cholesterol that the HDL that you make can't pick it all up, then that means there's going to be extra cholesterol just floating around in your blood, and then it can actually form plaques in your blood vessels, and that can lead to all sorts of problems in terms of atherosclerosis and coronary artery disease and those kinds of things. Now, in terms of using lipids for energy, lipids are great for storing long term because they're very easy to store, but they're difficult for us to use very rapidly because they are lipid soluble and not water soluble. Most of our enzymes are not super good at dealing with that. So breaking down the fat to use is much slower process, which is why we preferentially use carbohydrates. And the only way we can use lipids is if we don't have any carbohydrates that are available. And then your body's like, oh, dang it, I guess I'll use lipids, but I really would prefer sugar because it's easier. In other words, your cells are kind of lazy, right? You got to force them to do the hard work. So triglycerides can be broken down into their pieces, which would be the glycerol and then the 3 fatty acids. That process is called lipolysis. And then that glycerol can be converted to pyruvate, and pyruvate, of course, can then go into the citric acid cycle. It would be converted into acetyl-CoA and then actually bind up with enzymes in the citric acid cycle to extract more electrons and hydrogens, give off carbon dioxide, and all of the things that you already know about the citric acid cycle. Fatty acids actually are converted directly to acetyl-CoA, and that process is something called beta oxidation. It's kind of interesting. We don't really get into it too much over here, but if you look at this diagram, you can see here's our triglyceride. There's the glycerol with the 3 fatty acid chains. So the first thing that's going to happen is we're going to separate those. So now we have our glycerol, and that glycerol, you can see we're going to convert it to pyruvate. We actually get a little ATP when we do that. Not unsimilar to the whole process of glycolysis, and then that pyruvate will enter directly into the citric acid cycle. Those fatty acid chains, on the other hand, the process of beta oxidation is going to take each one of those chains, and you can see that we're going to take that fatty acid, we're going to convert it using coenzyme A into acetyl-CoA down here, and in the process of doing that, we're going to collect some electrons and hydrogens on FAD, and then now we have this, and we're going to do that process a couple of times, collecting quite a bit of electrons and hydrogens, but ultimately we end up breaking that fatty acid chain and getting the little bit of acetyl-CoA. You can see it's just a few atoms here where all of this is kind of waste, and then that acetyl-CoA will feed directly into the citric acid cycle. This whole process is called beta oxidation, and it's a great, this is why there's so much energy in fat, because you can see we're collecting a lot of extra electrons and hydrogens versus when we start with the carbohydrates. So, pros and cons to each process. All right, and then we have protein digestion. This one we really don't use unless we do not have carbohydrates or we do not have lipids. So we're talking about, kind of a famine situation here. The enzymes involved are going to be things like pepsin in the stomach and trypsin that comes from the small intestine. They are highly specific for particular peptide bonds in proteins, which is one of the reasons why proteins are hard to break down, and it takes more effort to break down proteins. So having a high protein diet, you are going to have a higher metabolism because you're going to have to invest ATP into breaking down that protein in order to get ATP. So it's one of those kinds of things. We can make amino acids. That's called catabolism. In order to do that, we have to deaminate. In order to break them down, we have to deaminate them, and then ultimately we'll convert them to pyruvate or acetyl-CoA. We can also make amino acids. That's synthesis. So breaking them down is catabolism. Making amino acids is synthesis. We can only make we can make a bunch of there's 20. We can make a bunch of them, but 9, I know it says 10 because your book says 10, but it's 9 are essential amino acids. Those ones we can't make, and we have to consume them in our diet. So I just always like to point out the deamination process, because in order to catabolize proteins, we have to remove the amino group from the amino acid. That's what makes an amino acid an amino acid. But the amino group has nitrogen and hydrogen in it, and if we're removing those, that means we're going to generate ammonium as a byproduct, and ammonium is super toxic. So your cells have a mechanism for getting rid of that toxic molecule, and that is the urea cycle. And so we can take that ammonium, and along with some carbon dioxide, we can convert it into urea, and then that urea we will excrete in our urine. obviously we wouldn't want urea building up. Or we certainly don't want ammonia building up. So anything that disrupts this system would be problematic. It would disrupt homeostasis. Now, that brings us to patterns of metabolic activity, because we can predict what your body will be doing at any given time. So we have two major states. We have the absorptive state and the post-absorptive state. The absorptive state is the time right after you eat. So if you eat 3 meals a day, then, for about 4 hours after each meal, you'll be in the absorptive state. So half the day, you're in the absorptive state. And that means insulin is going to be the major hormone that's sort of overseeing the absorptive state, because you're going to be focused on nutrient absorption and keeping blood sugar levels low, since you just ate a meal and you would be causing an increase in blood sugar levels. We want to immediately, use a negative feedback loop to pull those down, and that's insulin. If you haven't eaten recently, then you would be in the post-absorptive state, which is sort of like if you're sleeping, you're in the post-absorptive state. You probably spend at least 12 hours a day there. If you fast or you don't eat 3 meals a day, then you would spend more time in the post-absorptive state. And instead of having insulin here, we're going to have hormones like glucagon, but also epinephrine and glucocorticoids and growth hormone. Flashback to the endocrine chapter, because now we're going to have to rely on our energy reserves to meet our cells' energy demands. We don't have ready access to sugar in the blood since we didn't just eat. So instead, we're going to have to actually manage those reserves and release them on an as-needed basis. Okay, so I would be remiss if I didn't talk a little bit about nutrition. Vitamins, by the way, are organic molecules, and minerals are inorganic molecules. They are both very important. They both often are either coenzymes or cofactors in enzymatic reactions, which means if you don't have enough of that vitamin or mineral that acts as a coenzyme or a cofactor for that enzyme, then that enzyme can't do its job. And obviously, if an enzyme can't do its job, that is going to compromise the health of your cells and ultimately of you, the organism. So, vitamins, the organic molecules, can be divvied up into two groups: fat-soluble vitamins and water-soluble. So just take note, fat solubles are like A, D, E, and K. The water solubles are B and C vitamins. These ones are often coenzymes. Where the water soluble vitamins we often can absorb in the stomach, small intestine, but a lot of the fat soluble vitamins, we're going to need the large intestine and bacteria to help us absorb those. So that can be a little bit more challenging to make sure that we're getting enough of them. Important to note that vegetarians and vegans just have to watch the vitamin B12, because it really is only found in animal products or via a supplement. Those supplements are made from bacteria, which might violate your vegetarian or vegan, what it is you're trying to accomplish. But you do need to keep that in mind, because B12 is super important to red blood cell formation. Totally doable. You just got to be thoughtful and making sure that you're getting enough of it in your diet. Same for complete proteins. You want to make sure that you're eating complex complete proteins, and it's again completely doable on a vegetarian or vegan diet. You just need to do a little research and make sure that you're, combining the right kinds of proteins to get those complex or complete proteins, so that you get all of the essential amino acids that you need to be healthy. Most important thing, right, is we want to keep calories within a particular range. The kind of recommended calories on every item of food is like, a daily calorie intake of 2,000 calories. That's actually probably pretty high. Like many things, we base that on your typical male needs. And so women probably need quite a bit fewer calories than that. Some people who are more active or have more muscle may need more than that. So that really is just like a very rough estimate. And there is a lot of data, by the way, super depressing to me, but there is a lot of data that shows that people that restrict their caloric intake to like below, I think it's like 1,200 calories a day. I would starve to death. I would be so cranky. But people that really restrict their caloric intake actually like live significantly longer. And the research on this is quite robust. I'm not suggesting it. I love food, and I think you should enjoy good food. So, a great way to enjoy good food is the Mediterranean diet, and it's relatively healthy for you. It focuses on more plant and less meat products, which we know is good because it reduces heart and stroke issues and increases your lifespan. Adding, exercise, including weight training to help build bone. All of these things are what you need to really be at your maximum health level. And it's not easy. So, every day you try. Across the bottom, you can kind of see what you should be eating, and it kind of is like a visual representation of the volume. So you want, a good amount of grains, lots of vegetables, fruits, very small on the oils. You want calcium-rich foods, which most people get from dairy, although there are other ways to get calcium-rich food. But again, you'd have to really pay attention. And then proteins. And so grains and vegetables should be the bulk of your diet. A little bit smaller on the fruit, although fruit over dairy and proteins, So it's all about moderation. I wanted to just talk a little bit about how your appetite is regulated because I think it's really fascinating stuff. So here's our hypothalamus. So we're up in the brain. There's the pituitary gland, right? And it has a bunch of connections, your hypothalamus, to the stomach. So in terms, in the hypothalamus, you have a satiety center and you have a feeding center. And if we look at the satiety center, we're going to see that there are a number of things that will activate your satiety center and make you feel, kind of full, like you don't want any more. They're going to reduce your appetite. So things like elevated blood glucose levels should depress your appetite. Like you've already got a lot of sugar, you don't need any more. Whereas low blood glucose would stimulate your appetite center, right? Have the opposite effect. Also, lots of hormones from the digestive tract should stimulate your satiety center. Things like CCK, because if you're releasing CCK, it means you just ate a meal and you shouldn't want to eat more. And then also in your stomach, and all along the digestive tract, you have these stretch receptors that tell your brain like, hey, you just ate, so you really should feel full and satiated and not have an appetite right now. But it takes about 20 to 30 minutes to trigger all of these things from the time that you eat. So, you can eat a lot in 30 minutes. So this is the idea where if you eat slowly and you enjoy your food and maybe you're eating with people, which helps slow you down, that you'll have eaten less in that time that it takes to get your satiety center activated. Now, your feeding center or your appetite center, that is going to be activated by things like neuropeptide Y, and that will stimulate that feeding center and increase your appetite. But the one that I love is ghrelin because it just sounds like a little gremlin and I feel like it lives inside my bread. I'm hungry. So ghrelin is actually secreted by the lining, the mucosa of the stomach. And it stimulates your appetite. So ghrelin levels are high when the stomach is empty. And so it sends a message up to your brain saying like, I'm empty, feed me, feed me. Now, also interesting though, is that there's another hormone called leptin, and leptin is a hormone. It's a protein hormone, peptide hormone, that's actually released by adipose tissue. So the more adipose tissue you have, the more leptin hormone you release. And the idea is that leptin should suppress your ghrelin. So like if you have a lot of adipose tissue, it would suppress ghrelin so that you're not as hungry, because basically your fat cells are saying, hey man, we got plenty of fat. Like, why don't we just use up this fat instead of eating some more, right? I mean, this makes sense. There's just, and so we see that thin people have less leptin than larger people, which makes sense since adipose tissue is what produces the leptin. But there's a catch to leptin. So when you have too much leptin, like you're actually obese, not just like a little bit of extra fat, but medically obese, you develop a resistance to leptin. And so now you're producing the leptin, but it's not having the effect of making you feel satiated or suppressing your appetite. So now ghrelin is winning. See, I told you ghrelin's like a little gremlin. All right. So this is one of many issues that can make it really difficult for people to lose weight, because it is, of course, about the calories in versus calories out, but there's these hormones that are also impacting what your brain is thinking or feeling about nutrition and food and satiety and hunger. And that's challenging for your body. Okay, so the last thing I want to talk about are some clinical correlations. Anorexia nervosa and bulimia are real things. In anorexia nervosa, people just will not eat and they can actually starve. They can go into protein catabolism, which we know really isn't optimal. They'll enter ketosis, which is another thing that's not super great. When you start breaking down too much fat, it builds up ketones, and ketones can lower blood pH levels, which is, as you know from earlier learning, is not a great thing. Obesity is being 20% or more overweight. That's a little bit difficult because you do have to take into account muscle mass of the individual, bone size. So that 20%, it's a rough estimate. You really need to, you need to see the patient. It's not like a hard and fast number, but just roughly 20%. In the United States, like over a third of Americans are 20% or more overweight. So, and obesity does increase the risk of lots of other disorders and diseases. So there is a real motivation to try to not head into that. And it certainly does have impacts for healthcare, because it's going to create more, the more people that are obese, the more patients we're going to have, the more expensive health insurance will be. So there are some real implications to this. And it's probably definitely connected to lifestyle, at least in some part, but we can't ignore the physiological parts either. Okay, so high cholesterol is an issue that we're seeing because it's linked to coronary artery disease. High cholesterol is often also associated with obesity because that's linked to poor diet choices. We have phenylketonuria, or PKU. This is when you don't have a working gene for metabolizing phenylalanine, which is found in a lot of foods. And so if you eat things with phenylalanine, what happens is, because you can't metabolize it, it builds up and it leads to brain damage. But it's really easy. I shouldn't say it's really easy, because that's not really true. But if you don't include phenylalanine in your diet, then you won't have any problems due to phenylketonuria. So as a result, we test every baby born in a hospital in the US for PKU. So we know right away that they are not able to metabolize phenylalanine, and then you just eliminate that from the diet. Again, not an easy task because a lot of foods have phenylalanine, but it's doable, and it means that person can have a totally normal life. Kwashiorkor disease is when you get enough calories in your diet but not enough protein. This does also lead to brain damage and cognitive development issues. We see this in places suffering from famines. Ketoacidosis, I mentioned it earlier. If you start going after all of your fat, if you are not providing any carbohydrates for your body at all, then your body is going to go after its lipid reserves. But that fatty acid metabolism is not without problems, because your body does prefer carbohydrates. And the consequence of breaking down fatty acid is that you are going to create ketones, which are going to build up in the blood and ultimately lower the pH, and that can lead to a coma. It is not good. And this can also happen in diabetics because they're not regulating their blood sugars appropriately. Last one is gout. This is just very common, which is why I wanted to include it here. This is where uric acid builds up in joints. It means that we are not metabolizing our nucleic acids, in particular RNAs, correctly. And we're not going to get into any of the details of that metabolic breakdown of nucleic acids, but gout is not an uncommon disease. It can be connected to some diet choices, but it just seems to be maybe more like people have a predisposition to developing gout in middle or older age. Somebody who's very famous who had a lot of gout was Henry VIII. That was that king that had all the wives, many of whom he killed. Anyway, so gout is something you might see in a clinical setting. All right, guys, that's it for this. Hopefully this was helpful.